Every single day, your body sheds around 30 to 40,000 skin cells, and you never notice this because they are being replaced through cell division. So, what triggers a cell to divide and how does it pull that off? This whole process, from the time a cell is formed to the time it splits into two new cells, is called the cell cycle. So, let's break this down. Why does cell division via mitosis actually matter? Three big reasons. First, growth. A single fertilized cell called the zygote divides again and again, becoming an embryo, and then eventually a full grown human. You didn't get bigger because your cells got bigger. You got bigger because you made more of them. Secondly, tissue repair. Any damage to your body and the cells around it divide repeatedly to close the gap and helping with tissue repair. Finally, for some organisms like bacteria, division is reproduction. One cell simply splits into two, as you see here, and just like that, a new organism exists. That's asexual reproduction. The life of a cell, what we call the cell cycle, is a loop. It starts with the cell's formation from the division of a parent cell and ends with its own division into two daughter cells. This whole cycle can be broken down into two main phases: interphase and the mitotic phase. Interphase is where the cell grows and copies its DNA, getting everything ready for division. Then comes the mitotic phase, where division actually occurs. The cell's nucleus and cytoplasm divide, producing two identical daughter cells. But there's a lot happening inside each of those phases. So let's first start with interphase. Interphase makes up 90% of the cell cycle. All of this right here is interphase. And during this time, the cell performs its usual functions while also preparing for division. Interphase involves three sequential subphases: G1, followed by S, and finally the G2 phase. G1, or the first gap. This is the first stage of interphase in the life of a newly formed cell, where the cell grows and starts doubling its organelles. During the G1 stage, the cell is quite active as it is building up the raw materials and accumulating sufficient energy stores for the next phase. Now, the events of the cell cycle are directed by a cell cycle control system. Certain conditions must be met before a cell is allowed to advance to the next one. For example, proceeding from the G1 phase to S phase, it has to pass through a checkpoint. So what's a checkpoint? A checkpoint in the cell cycle is where a signal either allows the cell to move forward or stops it. The cell cycle is regulated via three checkpoints: G1 checkpoint, G2 checkpoint, and then there's this M checkpoint. The first checkpoint is the G1 checkpoint, which is towards the end of G1 phase. An analogy for the G1 checkpoint is the passport and document check at an airport. The officer checks the traveler's passport, ticket, and whether everything is in order before allowing them to continue forward. Similarly, at the G1 checkpoint, the cell is checked for whether it is large enough, has sufficient nutrients, and has suitable growth conditions available, and also that the DNA is not damaged. If these conditions aren't met, the cell won't progress into the next phase. And the cell pauses and attempts to fix the issue or exits into a resting G0 state, waiting until conditions improve. Once the cell clears the G1 checkpoint, it now enters the S phase, which is the synthesis phase, a period during which a cell replicates, meaning copies its DNA. Once copied, each chromosome has two identical strands called sister chromatids joined at the centromere. So this right here is an unduplicated chromosome. And once DNA replication takes place, each chromosome would have made a copy of itself, having two identical strands, and they're called sister chromatids. The cell now enters G2 phase, the second gap. Cell here continues to grow and makes the necessary preparations for mitosis, which is going to be the next phase in line. Some cell organelles are getting duplicated, proteins are being synthesized, and energy stores are topped up. The cell completes its final preparations for division during this phase. But before entering mitosis, the cell faces the second checkpoint, which is the G2 checkpoint. Again, that's towards the end of G2 phase. Again, size and energy stores are assessed, but the main focus here is making sure every chromosome has been fully replicated during the S phase and that the replicated DNA is not damaged. If problems are detected, the cycle halts again, giving the cell a chance to complete replication or to repair the damage. An analogy for the G2 checkpoint is the baggage X-ray inspection at an airport. The suitcase passes through a scanner where the officer carefully checks that everything is in place, there are no problems before you're allowed to move forward. Similarly, at the G2 checkpoint, the cell checks that the DNA replication has been completed correctly and that the DNA is not damaged before it enters mitosis. Once both these checkpoints are cleared, the cell is ready to move into the mitotic phase where cell division will actually take place. So let's look at the mitotic phase, which typically takes about 1 to 2 hours, a shorter window compared to everything else that the cell went through during interphase. During the mitotic phase, the cell completes two major processes. First, mitosis, where the nuclear material divides. Then comes cytokinesis, where the cytoplasm divides, producing two new cells. Mitosis is further divided into four major stages that take place after interphase and in the following order: prophase, metaphase, anaphase, and telophase, followed by cytokinesis, and that completes the process. A simple way to remember this order is using the acronym PMAT: prophase, metaphase, anaphase, and telophase. Prophase, the first phase of mitosis. During prophase, the loosely packed chromatin begins condensing tightly into chromosomes. The chromosomes now become visible under a microscope with its identical partner, the sister chromatids. Duplication, remember, took place during the S phase, but these sister chromatids actually become visible in the prophase stage. At the same time, the nuclear envelope, as you see here, starts breaking down. The nucleolus, the dark dense structure in the nucleus, also disappears. Meanwhile, the centrosomes begin to move to opposite poles of the cell and spindle fibers start emerging. Metaphase is the second stage of mitosis. The centrosomes are at opposite poles. The sister chromatids are along the midline, and each of these sister chromatids have spindle fibers attached to them from opposite ends. These sister chromatids are arranged along the midline of the cell, which is called the metaphase plate or the equatorial plate. At metaphase, imagine a tug of war. Sister chromatids are like the rope here, and the centromere is like that knot in the middle. Spindle fibers pull on the sister chromatids, but in metaphase, the tension is balanced, which keeps the sister chromatids lined up in the middle of the cell, ready for separation. But before this can happen, the cell passes through one last checkpoint, the M checkpoint. This checkpoint confirms that sister chromatids are properly attached to the spindle fibers coming from opposite poles, because the separation of sister chromatids during anaphase, which is the next phase in line, is an irreversible step. Think of the M checkpoint like the final boarding check at an airport. Before the gate opens, the staff makes sure everyone is in the right place and everything is properly secured. In the same way, at metaphase, the cell checks that every chromosome is correctly lined up and attached to the spindle fibers from the opposite sides, and only when everything is secure does the cell give the all clear, allowing the sister chromatids to separate and for anaphase to begin. During anaphase, the centromere splits as the spindle fibers are getting shorter and they're pulling on the sister chromatids to the opposite poles. So each sister chromatid is now pulled towards the pole its spindle is attached to. And this ensures that one chromatid in a pair ends up at each end of the cell and they become individual chromosomes. So at anaphase, the centromere splits like the knot snapping apart during a tug of war. The sister chromatids separate and they move to opposite ends of the cell. Telophase, the final stage of mitosis, and things start settling back down here. The chromosomes now have reached the opposite poles of the cell and they begin decondensing back into the loosely packed chromatin form. The nucleolus reappears and the nuclear envelope also starts reforming, creating two daughter nuclei. Now, at the same time, the mitotic spindle that was right here pulling on the sister chromatids, these spindle fibers start breaking down and they will be recycled by each daughter cell to help build its own cytoskeleton. So by the end of telophase, we have two nuclei that are fully formed. So why did the chromosomes condense in the first place and the nuclear envelope break down during prophase? The answer is the chromosomes condense so that they don't get tangled up when the sister chromatids start separating during anaphase. And the nuclear envelope also breaks down so that these sister chromatids can easily move to the opposite poles. They're not hindered by the presence of that nuclear envelope. Cytokinesis, the second major stage of the mitotic phase and the last part of the cell cycle. Cyto refers to cell while kinesis means motion. And cytokinesis ensures equal distribution of cytoplasm, producing two daughter cells that have exactly the same genetic material. Now, how this happens depends on the type of cell. In animal cells that don't have a cell wall, a contractile ring forms right at the point where the metaphase plate existed. And as this ring starts tightening up, it pulls the cell membrane inward, creating a crease called the cleavage furrow. And as the ring keeps contracting, the membrane keeps pinching off, eventually producing two daughter cells. In plant cells, however, they face a different challenge since they need to build an entirely new cell wall between the two daughter cells. Now, how does this happen? During telophase, vesicles are laid out right in the middle of the cell, and these vesicles eventually fuse from the center and start growing outwards until they merge with the existing cell wall, dividing the cell into two. Once cytokinesis is complete, two new cells are made. Not every cell follows the classic cell cycle pattern. Non-dividing cells may exit the cell cycle completely and enter a phase called the G0 phase. G0 represents a resting phase of the cell cycle. A cell that is in G0 is essentially inactive in the sense that it's no longer preparing to divide. For some cells, this could be temporary until they receive an external signal that triggers the cell to get back into G1 phase again. However, for others like mature cardiac muscle cells, neurons for example, this state is permanent. These cells rarely or never divide again. Now here's the full picture of mitosis from start to finish. It begins with prophase, chromosomes condense, metaphase where they align at the center of the cell, anaphase, cystochromatids are pulled apart to the opposite poles, and finally we have telophase producing two nuclei. The cell finishes dividing by cytokinesis into two daughter cells, and these cells now enter interphase again. And that's the full cell cycle from start to finish, with interphase preparing the cell for division, mitosis dividing the nuclear material, and cytokinesis completing the division into two identical daughter cells. S phase, remember, is important because it's here where the DNA gets copied so that the daughter cells that are made by the end of cytokinesis have exactly the same genetic material. Alright, this next part matters. It's the kind of image you'll run into under a microscope in the lab or on an exam. You'll likely prepare or observe cells from a root tip like an onion root, and since it's a rapidly growing part of the plant, many cells will be in different stages of mitosis. So use this image as your reference guide to help you identify the phase a cell is in. Interphase here, you'll see a solid intact nucleus, and then prophase where the chromosomes start becoming visible but not organized yet. By metaphase, they would have organized themselves at the metaphase plate. And then anaphase, you see them being pulled to the opposite ends of the cell. And in telophase, you can clearly see two separate nuclei. Now, take a look at these two root tip images. On the left, some cells are already labeled for you. You can see cells in metaphase. You can see a cell here in anaphase and a cell in telophase. You can also point out many other phases in this particular image. However, on the right here, you have the same root tip, but no labels this time. Pause the video and see how many phases you can identify. Watch the video again in case you have some doubts. And that wraps up this lesson on the cell cycle and mitosis. If it helped, drop a like, and if you haven't already, check out the Cell Organelles series linked down in the description. Thanks for sticking around, and I'll catch you in the next one.